And I'd like to start by thanking Ulf and Andrea in particular for inviting me to give this talk. I sort of come in from a slightly different angle. I'm a geneticist. Most of what we do are genetics and genomics. We've sort of come into the field of protein from a more systematics point of view. We're working with human populations, trying to understand human metabolism, the part of human metabolism that you can actually monitor and access through blood, through plasma and serum. So we actually started a project a long time ago looking at the genomics and genetics of a particular population in the northern part of Sweden that I'll tell you about. We went from genomics into transcriptomics, and then eventually we wanted to look at some of the protein profile of that population. And our first effort was not really working with Olink and working with this technology, but actually working with MS. And that was a sort of sobering experience in the sense that MS is a very powerful technology, but it's also a technology that takes a lot of time and a lot of processing time afterwards. So we had the cohort that I'll tell you about, which is about 1,000 people. We had blood samples taken, of course, and plasma samples processed through high throughput MS analyses. That was the biggest study at that point of a population that was done with MS technology. And at the end of the day, after a lot of processing, we actually were able to look at about 100 proteins coming out of that processing pipeline, which was interesting, and we got some nice results, but it was pretty disappointing if you want to scan the plasma proteome and you'd like to have more than 100 proteins to work with. So that was done before we went into this technology, and it sort of gives a perspective of what you can achieve, because with these technologies I'll show you, and as Ida said, 1 microliter, and with a few panels you can look at many hundreds of proteins actually and get very good data. So Ida has already told you a little bit about biomarkers, and of course if you sort of come in from an outside, you'd like to know, so what properties are you looking for when you're looking for biomarkers? And these are just some general points in terms of biomarker properties. One is, of course, you'd like this biomarker to be affected as little as possible by things that are not related to the disease in general. You'd like them to be measurable, of course, and you'd like them to be predictive of either incident disease, that is the woman or the man is actually have the disease, or something which has to do with prediction of future disease. So these are easy to put on the sort of slide like this, but if you start thinking about exactly what goes into biomarker identification and characterization, not all of these things are actually looked at. And in particular, we realized that a lot of the variance that is going into biomarker measurements may come from non-disease related factors. And how do you control for that? Well, the first thing is, of course, to measure that, to try to understand what are these factors and how much do they actually play into the measurements. So the study that we designed was really aimed to look at the protein profiles, and we took 2 profiling kits or panels, the cancer and the inflammation biomarker panels, and then we wanted to look at the measurements that we got out and try to relate them in a population sense to other measurements we had. In this case, we had a lot of genetics data for this particular population. We had measured all kinds of other sort of clinical quantitative measurements from blood samples, from clinical chemistry, and a lot of other measurements, hundreds and hundreds of measurements that we've sort of generated previously. And we have a lot of lifestyle information. I'll tell you about that in a minute, where we, by questionnaires and other ways, try to get a profile of these people from a lifestyle point of view. And of course, medication is important here. And the main overall purpose was then to see whether we could use all of this information to get our hands on the non-disease related variation in biomarker levels. And can we not only measure that, but could we also use that information in the future to control for this information, to get more precise biomarker measurements? And I'll tell you that in a minute. So the population sets that we worked with actually came out from a European study, which is called the Euroband study, which just stands for European Special Populations Research Network. It was established quite a few years ago as a more global health study of European populations mainly focusing on genetics at that time, but of course we wanted to bring in all the other measurements that we could. So it focused on some pretty unusual populations, and that's why I want to bring this slide up. Because at that time there had been, and there still is, a major effort on looking at cosmopolitan or metropolitan populations all over the world, in particular in big cities, or where you can get people to easily get into the clinic. We wanted to do something completely different, so we focused on 5 European populations that are really at the periphery of Europe in geographic sense, and also which have very different conditions in terms of their climate and in terms of their lifestyle and in terms of their food. And as you can see, we also were interested in the genetic risk factors. And of course, European populations do differ to some extent in terms of the genetics of background. So where did we end up? This is where we ended up in Europe. You can see essentially none of these is really in the middle of Europe. They're all at the very extremes. In Sweden, we were asked, so where do you want to put a population study where you can be so different or as different as possible from other European cohorts? And we ended up in Karesuando Soppero, which is villages, and actually a population on the edge of Sweden, very close to Finland, in the very northernmost part. And we collected it, or we focused there because we know that they have a climate which differs very much from the rest of Europe. It's subarctic climate. There's a lot of variation in daylight and temperature between seasons. The lifestyle of people living up there is quite different from most of Europe. The diet in particular, people eat a lot of game meat, reindeer, moose, and other game, and a lot of fish. A fair proportion of the population actually works outside most of the time. Part of them are from the Sami community, so they're reindeer herders. Many of them work outdoors for other reasons. And they're physically quite active compared to what you're doing right now, which is sitting most of the time. And we've also, from a genetics point of view, it's important to see this is a very stable population. People have grown up there. Many people have moved out. Very few have moved in. So from a genetics point of view, it's a very homogeneous population. And that goes for all the other ones. And the environment, well, there is a climate change going on, but it's not going so fast. So most of the time, we consider this as a very stable climate and environment. And if you look at the other populations from the rest of Europe, like the one in Orkney Islands, that's obviously a very local, isolated population with a very specific lifestyle. The one in the Netherlands, these are villages outside the main cities. So they're also, in the sense of being isolated in the Netherlands, they're isolated. There are villages from the Italian Alps, which are quite isolated, different villages in different valleys in Italy. And in Croatia, these were populations on the islands outside in the Adriatic Sea, which are some kind of isolation. So they're all sort of unique in the sense that they have different lifestyles and quite isolated. And of course, we didn't select these populations for this particular study. They were selected from a genetic standpoint that they would be very useful for mapping genetic risk factors in because of their homogeneity. But as it turns out, they're quite interesting. And for those of you who haven't been that far north, Karesuando is a very beautiful place if you like cold weather. And this is the daytime temperatures when we were having our collections up there. It's not always like this, but it's one of the coldest places in Sweden. So of course, climate has a major impact on people's living there in many ways. Light is another issue. So if you go up there like in, well, the winter season in particular in December and January, this is as much light as you get midday. It actually looks lighter than it is. The sun never really rises above the horizon. You get some clouds and you get some reflection, but that's about it. So of course, you know, there's a deprivation of vitamin D unless you supply yourself with vitamin D and so forth. So they're very special conditions. Now, so we selected these populations a long time ago, and in each of them there was a study conducted. So these are population-based cohorts. Everybody in each of these places were invited to participate across the ages from 15 up to 94. There was about 1,500 or 1,000 to 1,500 people in each place recruited. So the total study across Europe is about 5,000 people. And then we spend a considerable amount of time with each of these participants to try to do a health study as much as you can with a person in about 3 hours. You measure as many things as you can. Some of them are things you measure normally in the clinic, some of them here. We also went quite far in trying to measure bone density, lung function, and all the things that you can measure relatively easily without too much of instrumentation. And then, of course, we took blood samples to do all kinds of measurements in blood, including blood lipids and all stuff. And then we also had a long questionnaire that was filled in together with the nurse there in terms of their lifestyle. So, of course, occupation. We had a long questionnaire on the diet to get sort of as much as you can from a questionnaire, and activity, physical activity in free time, and as many things as you can measure within a reasonable amount of time. And we took DNA or samples for DNA to get as much out of that study as possible. I have one more slide in terms of the measurements that we took, just to give you an idea of more of what has gone on here. So clinically speaking, all the things you can measure on the person, and then the blood samples have gone through not only the classical lipids, LDL, HDL, and so forth, but we had a specific study in Germany where they measured over 160 different lipid fractions, all the kinds of lipid and lipid components you can think of in blood. We've had a study on glycans, also measured with MS to sort of take all the glycan fractions you can. We've looked at allergens of various kinds. And then, of course, down the line, you see this as proteins. So this is where we ended up, and we felt we have to do something more about proteins. And the questionnaire has all the diets. The medication is very important here. These are medications long-term and short-term, so we've coded them into medication codes, the ATC codes. Of course, we had the medical records in terms of what people had experienced in terms of diseases at that point. Sleep patterns is an interesting thing. We know that the daylight time is affecting sleep patterns, so there is a standardized questionnaire where you can sort of easily subdivide people into whether they have early morning risers or if they'd like to sleep in, and I'm sure you all know which person you are. But if you don't want to sort of classify yourself, you can fill in that questionnaire and tell from there where you are. Work patterns, family relationships, and all kinds of other things. So actually a long, long catalog of things to do. And in terms of the genetics, which was sort of our main focus at that point, we went through a generation of different technologies or that we worked on. So we started with SMP genotyping with the kind of Illumina chips that were available in those times, about 700,000 markers. Then we went on to exome sequencing to fill in all the variants. First with the exome chip, which has about 250,000 markers, and then we went to exome sequencing. And now we're at a stage where we are going to do some whole genome sequencing on subsets of the individuals. And from a genetics point of view, these cohorts are quite unique because, as I said, they're very stable, which means that when you talk to them, you realize that they're all one big population. They're like one big family in each of these populations. I'm not going to show you the pedigree, but the pedigree for Casecontrol is like 800 people in one family, which gives us the opportunity to sequence a few individuals and then impute out the rest. We don't have to sequence all the 1,000 people from that cohort to get all the data. So right now we're, with imputation and so forth, the genetics analysis I'll tell you about is based on about 4.8 million polymorphisms that we have information on in these particular cohorts. So it's a quite extensive list. But we had the proteins on here, and as I said, we didn't start with the Proseek study. We did start with the MS. I'm not going to tell you this. It's published in PNAS last year. And it was good, but it didn't really qualify as a sort of protein study in the sense that we wanted to do it. So we went ahead and looked around and ended up seeing this particular technology close by. Actually, Ulf Landegren and Mats, who was on this slide here, the inventors of some of this, was from the same department. So of course we knew about this development. I didn't know exactly how well it would work. So we've tried it, and we went ahead and initially took the cancer panel, which is the one I'll tell you about, and just applied it to the whole cohort of that we had from Casecontrol, the 1,000 people. And then we started to look at relationships among data. So if you start from a sort of epidemiological point of view, you have the biomarker data, and then you have all the epidemiological information. and you try to look for correlations there. So this is one way of looking at the relationships within the data from an epidemiological point of view. So on this study you have some of the proteins, and this is some of the correlations we're seeing, or covariates that we're finding. So on this side are sort of epidemiological covariates in terms of anthropomorphism, so age, length, and sex. And you can see each of them has a lot of correlations with, or covariates in terms of the biomarkers. If you look at the top here with the numbers, these are some of the medications. And each medication has a very sort of much narrow terms of correlation. So each medication may be correlated with a particular biomarker. But there are strong correlations, both in terms of the epidemiology, of the general anthropometrics and the medication. So out of the 77 proteins that we were looking at here, 55 of them were affected by epidemiological associations or covariates. And again, some of them influenced many proteins, and when we came to the medication, it was a very sharp and particular effects that we saw. So this is not surprising in a sense that we did expect epidemiological associations. I think there's quite a few of them though. And of course it just tells you that there's so much going on here except a disease maybe that affects these. So the next thing we started to do was actually look at the correlation, the co-correlations of measurements between biomarkers. So this is protein-protein correlations in terms of the numbers that we get out of the measurements. And you can see, again, I don't know exactly the cutoffs here, but the strength is indicated by the width of these lines between some of the measurements. And again, in some cases this is sort of expected that some of the measurements may measure parallel things in the same pipeline, but some of them were between molecules that had not been described in the literature to be correlated in blood like this. And the correlation coefficients are very high. If you look at some between Caspase-3 and EGF, 0.81, it's almost like a technical replicate. So of course, from a biomarker point of view, these things are telling us the same thing. You don't have to measure both of them. And again, many of these correlations were not known when you look into the literature. So some of them are between closely related molecules, obviously. Some are between things that at least we couldn't anticipate, and we didn't know exactly what to find here. So it's good to know that these things are not showing or measuring independent things in the body. Probably they're correlated in ways that we didn't know about. Okay, so the next step we took was to see, okay, let's put the genetics into this. Can we find genetic variation which is affecting the protein measurement? So what you do then is you do a GWAS, which is a genome-wide association study, using all these 4.8 million genetic markers, taking one protein measurement at a time to see, can we find any genetic correlate for that variation? And you end up with these Manhattan plots, just to explain to you. So here are the chromosomes, and the human chromosomes, and this is the p-value for association. And if you find something like this, it means you have a number of S and Ps in a particular place in the genome, in this case very close to the gene coding for this protein, where you find genetic variation affecting the circulating levels of these proteins. So each of this is one GWAS, but then we went through all the proteins, of course, and found that quite a few of these proteins are affected by genetic variation. So in general, genetic variation is affecting about 20% of the biomarkers, and in some cases, a very large fraction of the variation can be explained by these genetic variants. So in this case, it's up to almost 30%, almost 1/3 of the variation we're seeing between individuals can be explained by a single genetic variant occurring at some frequency. And in almost all of the cases, the variant that we found was located close to the gene encoding the protein, which means that this is probably just, or not just, but it is a variant which is affecting the expression level of that protein. But in some cases, a few cases, the gene where the variant was located was far away and not related functionally that we knew of to the protein. So here is, for example, the variant that we're finding is actually close to the ABO gene, the blood group gene, we're not measuring blood groups, of course. We're measuring something completely different. So you're seeing a functional sort of link between a genetic variant somewhere, in this case close to the ABO gene, and a downstream effect on some other biomarker. And there were one of those cases had been described in the literature, but a couple of the other ones had not been seen before. We don't know the functional sort of how it actually works from a functional point of view, but it's not the first case where your ABO blood group can actually affect downstream biomarker levels, which is sort of an interesting observation in itself. Okay, so we had a fair amount of genetics going on here. We have a fair amount of other biomarkers, covariates affecting. So then we said, oh, let's put all of this together to see if we take each biomarker and define how much of the variation in that biomarker in these population of 1,000 individuals can be explained by different factors. So the picture I'm showing you now is a bit complicated, but I'll walk you through it. So here on this scale is each of the biomarkers out of these 77 biomarkers that we had sufficient amount of data for. And then these different colors here indicate, the red color indicate how much of the variation, which is up here, the total amount of variation in that biomarker, which can be explained by different factors. So when it says top S&P, it essentially means genetics. So in this particular marker, if you just look at that for a moment, the first one here, we have 40% of the variation between individuals can be explained by one S&P in the genome. And then you can see when you go down here, the genetics decrease in importance. It still means a little bit, but it decreases over. On the other side, you have biomarkers which are influenced by other covariates. So in this particular case, we can see that it's highly influenced by H, which is the yellow one, and then you have the blue one, which is other things. So essentially, you can see we have one set of biomarkers which are very strongly affected by genetics, and the other one which is strongly affected by environmental covariates of various types. And then you have a small set of biomarkers in the middle here, which are to a much smaller extent affected by any of these variants. And I think we have, and of course from a sort of medical point of view, you'd like to find biomarkers which are as little as possible affected by either the genetic constitution of the individual or any measurable environmental factor. You'd like to have them be just affected by the disease, if possible. So of course, from that point of view, these biomarkers might be interesting. So then we said, okay, so these were some of the biomarkers, but the gray, if you look at the gray stuff up here, that's when we put all the covariates on top that we had. Almost 160 covariates, all the measurements, all the information we had from these individuals. We put them on top and see how much of the total amount of variation can we explain. And you see the gray bar is quite large, but this is then composed of many other things. And in general, we can see that we can explain in some cases up to, one, which is like here. That means we can explain almost all the variation we're seeing with the covariates we have. Now, this is not really true because we're using a statistical model which is not taking into account the correlation between these covariates. But if you look at the bottom part, which is harder to read, you can just see a gray bar here. That's when we combine the information, when we take into account that these are not independent covariates, they're actually correlated covariates. So how much of the variation can we then? Well, if you just roughly see the gray bar here, it sort of levels out at about 0.4 or maybe 0.5. That means that with all the information we have, we can still only explain about 50% of all the variation between individuals in these protein levels. And of course, the question is, what's the rest? We don't know. It could be technical variation. There could be other sources of variation that we don't know about. So these are all nice things, but what do you make out of it? How do you start using it? So the first thing we said, okay, well, let's look at some of these biomarkers in more detail and see what we can use out of it. So this is one example, the one to the very left in the previous, where we know genetics is very important. So this is a graph just to illustrate how strong the effect is. So the gray points here are the controls, are the homozygotes, and the red ones are the heterozygotes, and then we have a couple of blue, very few blue ones. And this is the correlation between the measurements on this scale and then the age of the individuals. So even though we have a sort of change of levels of this protein with age, you can see that depending on the genetics, you're always sort of divided away and above if you're homozygous for it. So genetics has a very strong effect, and it's plays out quite well across the age spectrum here. This is another way of illustrating it. We have different curves, the homozygote, the heterozygote, and the other homozygote. So again, this is the strongest genetic effect we're seeing. And if you think about it, 35% of the variation, that's a lot. And if you don't take this into account, and using it as a biomarkers in itself without knowing about these things, you can be quite inefficient and quite wrong. We see similar things. This is a non-genetic effect of medication. It doesn't look as strong because the curves are a bit on top of each other. And in this case, a smaller percentage, 4%, is can explained by the use of these drugs that affect the biomarker level in the blood. So we thought about, okay, so how do we use this information? It's nice to have to understand the different covariates and their effects. And we said, okay, let's try to develop a more personalized biomarker cutoff levels. So we introduced this concept, which we called personally normalized plasma protein profiles. And what do we mean by that? Well, this is more of a statistical term in the sense, but we try to model the effect of all these covariates that we have then measured and quantified in their effect sizes on the protein abundance. And essentially, the idea is to, knowing this information for a particular individual that comes in and has a measurement done, we also collect information on the covariates. Some of them are quite easy to collect because you ask about the age, sex, and so forth, and all of the ones. And for each protein, we know which covariates are important to collect information from. And then we introduce that covariate information and try to develop a cutoff levels, normal levels for that person, given his or her covariate exposure. And then try to see whether this can be used to have a better use of the biomarkers. That is, that we can discriminate better between people that have disease and those that are healthy. And we've tried to use this in our cohort, given the fact that the cohort is obviously a healthy cohort, but of course this is a population-based study, so we do have people who have suffered from different diseases. So we pulled out, in this case, groups of patients that have had cataract, diabetes, hypertension, myocardial infarction, and stroke out of our own data, and took those aside, developed what I went through here, developed the covariate profile for healthy people first, and then used that covariate profile on the different disease groups to see whether it would actually give us a better discrimination in terms of the biomarker levels between those that are healthy and those that are sick. I'm going to give you a couple of slides on that, and again, a fairly complicated slide, but it's not so hard to understand. So let's take one thing at a time. So here is the group with cataract. So on this side, you're seeing the number of significant associations, that is, the number of significant associations with biomarkers. And we have two sort of colors here. You have the red one, when we've just taken the biomarker data without using this personalized, normalized levels, without taking into account all the covariates that exist and that could affect it. And you can see in this case, we're finding for this particular diseases about 80 different proteins or associations, potential biomarkers if you wish. But once you start to use this particular method, once you take into account the covariates that we know are important for individual proteins, this is reduced down to less than 4 or less than 10. So we're reducing the number of interesting candidate biomarkers by taking into account on an individual level the types of covariates that exist. So these are the number of associations. These were the number of proteins. The staples or height here is, so there were like 35 proteins initially, and then it came down to less than 10 when we adjusted for on an individual level, not on a population level, but individual level. And the same story goes for the diabetes, high blood pressure, myocardial infarction, and stroke. We are reducing the number of interesting biomarkers to look for dramatically in both cases. And why is this important? Well, it's important because if you're going to spend time on a biomarker doing follow-up studies, you'd like to pick, and you'd like to be very picky about which you pick, because there's a lot of effort goes into each biomarker. So this is the one thing, or one way of looking at it. And then we did a similar but also quite interesting analysis where we try to see, so given that we have information on the different biomarkers and different covariates, how does it play into the number of significant differences in a case control comparison? So again, it's a fairly complicated picture to look at initially, but it's not so hard. So again, here we have the diseases, 1, 2, 3, 4, 5, that came out of the cohort. So on this side in red, you have the measurements, and in each of these line is actually one of the proteins that we looked at. And on this side, you have the measurements based on the raw data, meaning data where we haven't adjusted for all the covariates which we know play into each protein. And on your left side in blue, you see, left side in blue are the ones which are uncorrected, the raw values not taking into account the covariates. And on your right side here in red is the ones whence we have corrected for all the covariates that we know play into each. And when it's dark, it means there is a significant difference between cases, that is patients, and the controls. And the take-home message is quite simple. You see that on your left side, when we use the raw data, you have a lot of statistical significances coming out. And when you look on your right side, where we have the corrected values, there are very few. Again, so you're taking into account the background variation and reducing the number of spurious significances, leaving you with fewer comparisons, fewer proteins that are of interest to examine further. And independent of which of these disease or category of patients you look at, you see the same thing. You see here's a lot of ones that fall out, which looks very, if you don't take into account the covariates, look very interesting. There's strong effects, significant. But when you take into account all the background covariates, they all disappear. And there are a few that are left here. So we think this is an interesting way of refining your measurements in terms of finding biomarkers and reducing the efforts. But it also means that you can probably faster move into the clinic with some of these things by getting what we call linear cutoffs. Because from a clinical point of view, you'd like to take a measurement and know that if the measurement is above or below a certain level, this is something to be aware of and something to do something about. But if you have a lot of background correlations with age, with other covariates, you have to take those into account, and the cutoff becomes like an age-dependent cutoff. So our belief is that if you know which biomarkers need to be adjusted by which covariates, you can do that, and then you can end up with similar, with linear cutoffs. So this is just one example. This is the protein, and this is the patient category, myocardial infarction. So the dashed curves up here are the measurements for the raw data when we have the controls and we have the cases. And you can see that in general, the cases are in red and the controls are in blue, in case you can see it. So you can see in general the cases have across the the weight spectrum here, you have a slightly higher value, and then at one point they cross each other, which essentially means that, yeah, this could be used in the clinic, but when people become overweight over here, you have to take that into account to evaluate your measurement. So these are after we've adjusted for all the covariates. You can see that there is the cases are all across the whole weight spectrum. They're always above the controls, which makes it easier, which makes it essentially possible to put a clinical cutoff right here, and that goes across all the covariates, all the different differences between patients that may exist in other respects. And similar things, sometimes if we adjust for all the covariates, there is no effect anymore. And it means that you cannot use this biomarker even, because if you look at it from up here, these are the uncorrected values. It looks a little dippy up and down, but it looks like the cases are always above the controls, and it could be useful. But when you take into account the fact that people, different weights, blood pressures, blah, 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 and they take different medications, really, this is not a good biomarker. So you can put that aside and say it cannot be used. And things can be complicated. This is a GDF, growth factor. This is the uncorrected values. Again, it looks like these could be a good value, but it dips up and down a bit. Here we have, when we adjust for the covariates, you can see that for the controls, it's a flat line essentially. And the cases are always above, except in this particular point, which we don't really know. So it sort of illustrates the fact that we think that we can use these biomarker much better if we take into account the covariate variation in an individual level. So just to try to summarize here, what have we learned so far? Well, I think we've learned that genetics actually play a much bigger role than we've thought, and that in general we have a lot of variation in these biomarker measurements, which is unrelated to disease. If you want to use that information, well, we probably need to do some genotyping of people if we want to put them in the right category from a biomarker point of view. We think that the method we have developed seems quite useful for identifying among from a screening point of view, the top set of biomarkers to be evaluated further. And again, then for prioritizing among these candidates for clinical validation. And it tells you that the biomarker studies really have a long way to go, I would say, because most of the biomarkers that Ida was pointing to here that are coming up are not entering the clinic. And of course, there are many reasons for that, but I think one of the reasons is that most biomarkers have not gone through this kind of validation. We know a little bit about which covariates can be important, but they've certainly not been scrutinized in this kind of way. And it would be so much easier if there was a general pipeline that biomarkers that came out also went through this kind of validation. We could at least point to the particular covariates which are important, and we have a method to deal with it. It's not impossible to do it. It's just a statistical kind of analysis to do. And with that, I'd just like to point to some of the many people, my own group over here in Stockholm, in Uppsala. We worked with the hospital, of course. We worked with you at Olink. We worked with the SciLife clinical diagnostics platform where the assay was run, and the National Genomics Infrastructure, which is helping us with some of the genomics analysis here. So with that, I think I'm